Good afternoon, everybody. My name is Yaize Ontoria. I'm a PhD student from University of Barcelona. And today I'm going to talk about the effects of temperature and salinity on the functional traits and photosynthesis-irradiance relationship of the seagrass Halophila ovalis. This is part of my work that I did last year during my internship in Ecology University with Catherine McMahon. So, summer 2013-2014, Southwestern Australia, an exclusive event took place in this area. A mortality of Halophila ovalis was found in lesional estuary. And this day of event during this summer got worried people about this situation. People were asking or looking for answers about what could be the reason of that or what could happen in this moment. So there were some predictions, and one of them was that maybe the hypersalinity event that was recorded during this year there, or maybe the warming event, could be one of the reasons. So let's see how these two factors, hypersalinity and warming, act in this plant. We know that global climate change provides a number of threats to coastal ecosystems, and in this diagram, we can see all the climate drivers that act in the southwestern Australia estuaries. There are some climate drivers, but we are going to focus only on two of them, water and air temperature and rainfall. So we know that salinity of these environments will be rise through evaporation, and these are the two stressors that we are going to test here, water temperature and salinity. So seagrass ecosystems are particularly vulnerable to the effects of climate change, and we also know that shifts in salinity and temperature can have effects on metabolism rates, nutrient uptakes, survival, between others. And seagrass in estuaries are subjected to natural changes in environmental conditions, which are repeated at different temporal and spatial scales. So depending on the persistence and intensity of these changes in these environments, plants can be exposed to an stress event. And to cope with that, plants can develop two kinds of strategies, the adaptive ones and the non-adaptive ones. The first ones are related with the plant resistance, and the second ones are related with the deleterious effect of the plant. So in this estuaries of southwestern Australia that have fluctuation in light, salinity, and temperature, they can grow only some species of seagrasses, so some of Zostera, Rupia, and Halophila. And particularly Halophila ovalis has the widest range of environments, and this is the species that we are going to talk about today. So, the aim of this study is to evaluate the response of Halophila ovalis to the effects of warming and hypersalinity. And the three of our hypotheses are: first, that warming will affect plant performance negatively; second, hypersalinity will affect plant performance negatively too; and also warming and hypersalinity will act synergistically, leading to severe consequences for seagrasses. And to test that, we did two experiments. The first one aims to assess if the photosynthesis-irradiance relationship varies with temperature and salinity. And to do that, we choose a range of temperatures within the thermal range of this species, and also we reach 34 degrees, trying to know what will happen if a heat wave takes place. And also we choose two different salinities, the control one, 34 ppt, and highest one, 42 ppt. At the end of the experiment, we got the PR curves, and from them we got the net photosynthetic rate and the respiration rate. And after that, we did a second experiment. This experiment aims to explore the effects of the warming, the temperature, and the salinity on the plant functional traits. So we choose two temperatures, the control one 24 degrees and the highest one 30 degrees. And two salinities, the control one 34 degrees, ppt, and the highest one 40 ppt. And after a progressive increase of these two factors, we kept the plants during 10 days in an acclimation period, And after that, we kept the plants in a short-term experiment for 13 days under this stressful condition. At the end of the experiment, we measure the plant responses at physiological level and also an individual level. And also we measure carbohydrates. So I'm going to talk now about the results of the experiment 1. We got all these PI curves for the control salinity and for the high salinity. And if we look at both plots at the same time, we see that at all temperatures and for the two salinities, all the photosynthesis-irradiance relationship follow the typical pattern, and no photoinhibition was found. But if we look only at this plot, the control salinity, we see that we found the highest photosynthetic rate at lowest temperature, 20 degrees. And however, if we look on the right, we found the highest photosynthetic rate at highest temperature, 34 and 31. From this PI curve, we got the photosynthetic rate and also the respiration rate. In this graph, the response variable on the y-axis and the thermal treatment on the x-axis. And the colors indicate the blue one, the control salinity, and the green one, the high salinity. So looking at on the left, we see that there is an effect of the temperature. In the control salinity, the blue one, the temperature decreased the photosynthetic rate up to 28 degrees, but then increase again at 31 and 34. And also, if we look only at the high temperature, high salinity, the green one, we see that photosynthetic rate increased with the temperature. But if we look to the interaction, we saw that above 28 degrees, we found a synergistic effect between these two stressors. If we look at the respiration plot, we see there's only an effect of the temperature, since the respiration increased with the temperature above 28 degrees. Looking at the results from the experiment 2, where we kept the plants under stressful conditions, we got first the results from at physiological level. We measure maximum quantum yield and maximum electron transport rate. The colors are the same. The blue one is the control salinity and the green one is the high salinity. And we see on the left that maximum quantum yield was affected by temperature, since we saw a decrease with high temperature and not effect of the salinity was found. And looking at the graph on the right, we saw also an effect of the temperature, since the maximum ETR increased with the temperature, but not effect of the salinity was found. And at the individual level, we saw that plant growth was affected by temperature. The plant growth decreased with high temperature, but there is no effect of the salinity. And looking at the shoot survival, we saw that there is no effect of the temperature, and however, there is an effect of salinity. Salinity seems to increase the shoot survival at any temperature. And at the end, we measured the carbohydrate content, and we saw the carbohydrates decrease with a high temperature and not effect of the salinity was found here. To summarize the results, we can see that warming increased the respiration rates and decreased the photosynthetic rate, and also decreased the photosynthetic efficiency of the plant. And at individual level, warming decreased plant growth. However, hypersalinity increased the photosynthesis rate only at high temperatures, and also hypersalinity increased the survival. So from these results, we could say that interaction between increased temperature and salinity were not found at the individual level. However, a synergistic interaction was found on photosynthetic process. Also, high salinity seems to mitigate high temperature effects. It does not fit up into the next plant scale level, since we still observe effects of the high temperature in plant growth. And we could conclude that high temperatures are likely to have contributed to the die-off of Halophila ovalis in southwestern Australia estuaries, but there is no evidence of a negative effect of high salinity on functional traits and survival. But we still have some questions to answer. 40 42 ppg is not a stress factor for this Halophila ovalis population, but we will need to know which is the tolerance of this species. Also, what would be the response of Halophila ovalis in a long-term exposure, because we have done this only in a short-term experiment. And in the case that this species can adapt to these changes in the environments, in these environments, which are the mechanisms of adaptation. So thank you for your attention, and also I would like to thank you all my colleagues for the help doing all this work.
Any questions?
Can I ask you what is their salinity level at the site when you collect the sample?
34, around 34, because we choose the control one, 34, like when we measure in the field, we say, okay, this is the control one.